So I work in the Morgan lab here at UCSF. We're a biochemistry lab, and we focus on questions related to the cell cycle. So the process of cell division, mitosis. I work specifically with the protein involved in chromosome segregation. And so basically my job is to use these proteins in different biochemical assays to be able to evaluate their activity and characterize them better. And so in order to do these assays, we need a large amount of these proteins. And so to express them, we use both bacteria and insect cells. And to express proteins in insect cells, we use what's called the baculovirus expression system. And so basically this expression system exploits viruses. So viruses are dangerous and harmful to us and our cells because they're able to hijack our cells' machinery to produce in mass like their own proteins. And so we've been able to use that to our advantage by using molecular cloning to incorporate genes that encode for the proteins that we want into the viral DNA. So then we're able to infect insect cells with these viruses that are coding for the proteins that we want. And so that the viruses in those insect cells will mass produce those proteins. And so after we harvest those cells that are expressing these proteins, we then need to purify them with a system called the ACTA. And basically the ACTA has a bunch of different protein affinity chromatography columns. And so these columns are basically filled with a bunch of beads that have molecules attached to them that will bind to a specific tag that we've encoded with the protein that we have produced through the baculovirus expression system. So we're able to catch the proteins that we want using their tags on these beads that are in the column. And then we're able to elute them with a competing molecule and an elution buffer that allows us to then extract the protein off of these beads. And so now we have the purified protein that we're then able to include in biochemical assays. And we're able to evaluate the activity of the proteins, how they interact with other proteins. And that just helps us get a better understanding of how the process of cell division works. And this is helpful because it could potentially be informative for trying to understand disease models that are related to proliferative cell growth that is caused by unregulated chromosome segregation. And it also is just helpful in potentially thinking about how we could exploit the processes of the cell and apply that to translational research in medicine. Yeah, so that's basically what I've been working on, kind of the gist of it.